All right, sorry about that. I had to get the stream going properly. And so our last chapter this week is polymers, and then a little bit of biochemistry, which does get into polymers as well. And so I'm going to save the demo for Thursday, but let's get into kind of what are polymers. And so I put up some examples of polymers, but let's just define them. And so polymers are just repeated, or a polymerization reaction is repeated addition of small molecules to form a long continuous chain. And then monomers are just defined as their small units from which our polymers are synthesized. And so I put just like a very, generic monomer polymer visual here. So of course, right, this would be our monomer unit, and then we link them together to get our polymer. And so I showed a few polymers here. And so of course, DNA is a polymer of all our amino acids. And then nylon was also a polymer that was synthesized. So a lot of your synthetic fabrics, right, are polymers and synthetic as well. And so let's look in at our polymerization reactions a little bit. And so if we look at our polymerization, a true polymerization is a radical mechanism for the most part. So true polymerization is usually a radical, but then we can also have kind of other types of polymerizations, but I usually call them more like cross-linking polymerization. And we'll talk about those as well. But cross-linking polymerization tends to involve condensation reactions. And so, again, most of these polymerizations are radical mechanisms, and so we can just appreciate the mechanism here. And so this is ethylene being polymerized. And so if we look at our mechanism, so our radical, so they have it as an hydroxide radical. It can just be, any radical. And so if you were to get into atmospheric chemistry, by the way, there's a lot of radicals in atmospheric chemistry. But then our radical, so our single bond is safe, but then our double bond, right, in our pi system, so it's the electron in our pi system, is not. So then our radical will kind of attack this bond, and so then we can make, right, a new bond, so with our radical, and CH, H, and then this splits, and so then, right, this carbon now becomes a radical, and so then it can continue on and on and on with our polymerization, and then eventually, and it mentions it here, so your termination occurs when two radical ends meet, and then your polymerization is over. And so we won't get into all the details of this, but basically, right, you can take a whole course on polymerization and get into all the conditions and how you select for certain lengths of chains. So that's what you want to be able to do in industry, right? Do a reaction, have them all come out with around the same length of your polymer chains that you're looking for, etc. So all that can be optimized by whatever reaction conditions. And so other things to note are just kind of what our monomer is versus our polymer. So here's our polyethylene monomer. So of course this is ethylene with our double bond. And then the polymer, again, right, that double bond just becomes a single bond from that radical mechanism and then just repeated. And so just note when we look at these polymers, right, then they kind of put all this in parentheses. So this is to show our monomer unit. And then our N here would just be however many times it repeats. So again, if we were, doing industry reactions, trying to optimize, then we'd probably have a number for this N, but right now we're just kind of looking at the generic basics. And so just continuing with other polymers, so we have Teflon. I think you had this one in lab. And again, right, notice the double bond is susceptible to that radical mechanism attack. And then our monomer is just our single bond repeating. Questions so far? And so then your book has this really nice chart that I just kind of stole. And again, this is just so we can appreciate the monomer and then, right, see the corresponding polymer. And so, of course, we have PET material. And so this is pretty common. So I think this is either 2 and 4 in the plastic recycling. I have to double check my numbers. But we have polyethylene, and this can be either high-density polyethylene or low-density polyethylene, depending on the branching. But they're both made of the ethylene monomer, of course. And then structural unit is just, of course, our 2 carbons, which can repeat. And like I said, again, branch if it's low density. And so this is probably the most common plastic that you see. And so it can be, right, anything, toys, buckets, containers. I mean, we really have gone crazy with plastic. I mean, that's something else we can talk about is just plastic and microplastics and how they're pretty difficult to break down and recycle. And then we have polypropylene, of course. So here is our polypropylene unit with our ethylene and then our methyl group hanging off. And so polypropylene, again, more food packaging, clothing, so really everywhere. It just depends on like the different properties, what you're going for, and like how hard they get. So depending like, at what stage you blow them, and then like they're appropriate for different applications. Yeah, so I've actually not. Sure, yeah, so I think these molded plastic parts, the polypropylene, I think they can come in like smaller molds, and then in the factory they kind of blow them up with heat, and then, they're the appropriate size, like sriracha bottles, for example, or, any plastic manufacturer. And then, so PVC, or our vinyl chloride, so that gives, of course, that like harder plastic that you see in like water tubing or pipes, other building materials. And this is a nice new polymer, so neoprene. This is often good if people have like allergies to latex, which is pretty uncommon, or not, sorry. Latex allergies are pretty common, but nitrile allergies are uncommon. And so if you are allergic to nitrile, though, then you can try neoprene. And so that's supposed to be better. So this is a different polymer. And so it mentions other uses, so hoses, wetsuits, gaskets. And of course, that's the other thing, too, is, right, they're trying to develop new materials in industry, trying to, tune your monomer to give you slightly different properties for your plastic that you're interested in. And so this is a cool one. I wouldn't like commit this to memory or anything. We can just appreciate it. And so this methacrylate monomer, so this is used to make plexiglass or other like visors and shields. So it probably has a good property, right, of letting light in. So helmet visors, aquariums, contact lenses, so lots of things with that polymer. And so then we have Teflon. So this is used in a lot of non-stick coatings. Personally, I would stay away from Teflon since I think they can react and kind of burn off anyways. So I don't know how safe Teflon is, but I still use it. It's just that non-stick coating, like, you know, like, yeah, like, have you seen a non-stick pan? And then it's got like that, and like it feels slick, right? Yeah, so that's, at least, right, if it says Teflon, then it's probably a Teflon coating. Yeah, personally, I don't trust anything other than a cast iron pan, but that's another rabbit hole. Yeah. Pure. I don't trust anything coated. And then acrylonitrile, or acrylonitrile, sorry. So this is acrylic fibers, so tents. Again, this is kind of interesting, right? It has that cyano group on that. And then this is one we have mentioned before, somewhat important. I don't think I'm going to ask you guys trivia of polymers or anything, but I would at least kind of be somewhat familiar with styrofoam. And so styrofoam has this styrene, or right, this benzene ring basically on it. But this is also why styrofoam is difficult to recycle, because it has this styrene ring, so it can't really be made into anything else. Oh, yeah. And so kind of again in general, so right, all these polymers as well, so these are made from, you know, crude oil materials, so they're all hydrocarbons. And so, right, if you're thinking of, you know, how to be green, how to recycle, so of course the big barriers though to actually recycling is trying to break up these monomer units, or these polymer units, sorry. So once you make your polymerization reaction, so this is a pretty sturdy reaction, right? Doing that radicalization, now you've made these strong bonds. Obviously, right, these strong bonds are reflective in the materials of plastic. But then the problem is, well, how do you break those bonds back down? So that's really the biggest hurdle of recycling, is just the amount of energy that you would have to put into the polymer material to then break up that polymer and make monomers again. And so even if you can overcome the energy argument, so right, you can invest the energy into recycling, the next difficulty is, of course, the plastics have to be clean to recycle. And so, right, we all know that. We get our soda bottles and they all look really nice and clean plastic. And so the problem with that, in order to make nice and clean plastic, you have to start from nice, clean materials. And so it's, again, usually not worth it to get the plastic clean and make sure that it's, you know, actually pure enough to be used, again, to recycle. Because, again, you don't want to have cloudy plastic, you know, at least from the consumer perspective, so it has to be really clean. And so that being said, right, that's why it's, again, difficult to recycle, doesn't happen. I mean, even here in Oregon, what do they just collect with the recycling? Isn't it just one, like the bottles, you know what I'm talking about at home? Like, they don't do very much recycling.
Maybe told me that it goes to 2, but that's about it.
Yeah, I forgot, even though I should know.
But that would be an actual recycling.
Yeah, because y'all live in Medford. What does a road disposal pick up in the little bins? Is it just one?
I believe they pick up 2 or 3 different types. I did a little tour of the place like 2 or 3 years ago, and you'd be surprised they actually do a lot of recycling thing.
Yeah.
But they're also like, through the tour, they talk about like the industrial difficulties of separating the types of plastics and all that, because they have to do that at road disposal. Because, you know, people don't do it, you just throw it in the recycle bin. It's not separated by the type of plastics or anything like that.
Yeah.
Uh,
I don't know. It'd be interesting to get an updated tour, because the other thing is, I know like years before, yeah, and I'm even thinking like years before you went, so like prior to COVID, you know, other countries would purchase our trash and our plastics to recycle, and that's just become less and less profitable, so people don't do it as much, so.
On the website, it says just plastics 1 and
Okay, so yeah, so I thought it was even less than that, but that's somewhat encouraging. So right, rogue disposal does 1 and 2, but that's still pretty limited. So again, you can kind of get a sense of that.
Also, one thing to push them is they do the CNG. They use the natural gas off the landfills for fuel for their trucks.
Yeah,
It's pretty cool. And they use it for the public transport buses too.
yeah. Yeah, so that's something just to keep in mind. And then, yeah, it's good that like people are doing other things then to kind of offset our carbon footprint. Like,
I thought that was kind of cool, collecting like the gas from the trash and landfill and using that as food.
yeah. And I mean, this is probably a debate for another day, but it's like, should we go back to paper, you know? So paper products, we kind of went from paper to plastic, but who knows if that was really a better choice, but. I mean, there's a lot of questions of maybe we should just do like glass, you know, where the milkman and then you had to like keep trading back your glass, like maybe that promoted conservation and recycling more, so. I don't know, there's a lot of like philosophical things we could get into. Yeah, and what actually influences people to recycle. All right, so we'll get off our philosophical topic and let's look at some reactions. And so now let's look at a polymerization reaction, but now doing a condensation. And so again, these aren't radical catalyzed now, but instead, so a condensation reaction. And so kind of think back to, I think, our last chapter, right? So we said removing H2O, so that was a condensation. And so usually, right, when you do your condensation reaction, the reason why you're removing water is because you're making a new bond, so water has to leave. You can also think about other things that can leave. So sometimes, right, you have like acid as a leaving group or some sort of alcohol. And so let's look at this. So we want to be able to piece together how they come together. So this could be on an exam. And so we have our carboxylic acid to just kind of identify those groups. So carboxylic acid. And so, right, maybe we're somewhat learning that the carboxylic acid group, so these are susceptible to attack by something with electrons. And so that's what's going to happen here, is our nitrogen's going to attack here. And in the process, we won't go through the mechanism, but you can see, right, new bond forms. So now we have an amide bond formation. So that's our carbon-nitrogen bond there being formed. And then again, water leaving, and so everything's nice and colored, so you can kind of tell. And so like I said, on Thursday, I'll bring in this demo so we can actually do the nylon synthesis. And so again, you just want to be able to recognize where they're going to meet and chain up. And so this is also would be like a copolymer, since it's made. So let's skip down. So I have our definition of a copolymer. So that's just a polymer made from more than one type of monomer. And so going back to our nylon, and this is actually nylon, we should name this properly, 6,6 that we're doing. And so it's called 6,6 for the hexane chain that it has on both of these monomer parts. So 1, 2, 3, 4, 5, 6. And so this is hexane diamine. So with our 2 amines on the end, so right, it's ready to pair up with, in this case, our adipic acid. So And again, we can see where they're going to attach. So, right, our nitrogen will go here, and then another chain can add to this side. And so I have the polymer shown out. So right here's our two different monomers that went in there. So of course, right, we can identify the hexane diamine and then our adipic acid. And then note, right, we just have our monomer units. So one of our hexane diamines, one adipic acid, and then of course, right, we know it would go on and on and on. So C6H12 and H, right, etc. So just be kind of aware of that. So we know how to chain them. And so this might come up, just this terminology of, again, our amide bond. So that's important in this chapter. So we went over our copolymer definition. And so now here's another reaction. So this is polyester. And so this is an esterification reaction. And so esterification, do we remember what that was from last week? Yes, so condensation reaction. And so what's an ester?
It's oxygen. It's like the carboxylic acid group without the H, and it's attached to some carbon chain inside.
Excellent. So we make some sort of ester group. And so this polyester then, so we can write back out what it means. So right, just like that ester, so to make those fun bananas compounds and other things. So we added an alcohol plus our carboxylic acid to indeed make the ester. So that was good. And so this is just an example of our polyester shown here. So again, we have our alcohol. Notice it has an OH on both sides, right? So we can attach and keep going because it has to be able to attach to another side of our ester, right? So the other side of our carboxylic acid. And so again, kind of note that on the polymer, there's structures, right? So they have to kind of be able to attach at both ends. So this has to be in the middle. Again, alcohol groups on both sides to do that. Same thing with our carboxylic acid, right? So you can kind of think back. Actually, let's just flip back. Right, flip back to the notes of last week. If I can find it. So right, we had gone through our esterification reaction, so alcohol plus carboxylic acid to make our ester. But of course, that was just one, right? So we had an alcohol on one side. So we can see that bond formation. And so now to compare it, right, it just means that it has our carboxylic acid group and alcohols on both sides of our monomers. So then, again, it can add and keep going and going and going. And so we could walk through, right, the mechanism, but basically this is susceptible to attack. So like we had gone over before, the alcohol group is our new O that attaches, right, as part of our ester, and our original OH leaves. And so it's kind of shown here, right, that this is our water that's going to be leaving, and then new bond formation with this oxygen and our carbon. So that's shown here. So kind of highlight it, right, these are now over here. And then, of course, right, our alcohol is at the end, so it can keep going and reacting with another carboxylic acid. And then again, we want to just identify our monomers. So we have one monomer, right, our other monomer, and then of course our whole kind of polymer monomer unit there. since that, so basically, right, our copolymer unit would be this whole thing. Does that kind of make sense? And so something on an exam too that you might be given would, it might give you two structures of like an alcohol or carboxylic acid, or maybe, right, looking at our nylon, an amine, and a carboxylic acid, and then it's going to ask, what's the polymer unit look like? So that sort of thing would be on your exam.
So it is always the carboxylic acid that lose a OH group.
Yep. Yeah. So, and that's just something that we've seen in our mechanism. So this carboxylic acid, it's susceptible to that electrophilic attack, and then yeah, that means that it has to have some sort of leaving group there. And I think in other versions of this reaction, of this nylon synthesis, you can do it with another compound. And I think it has like a chlorine group there that leaves instead. And so just more things about plastic. So just kind of appreciating that, oh yeah, a lot of raw material goes into plastic making. And so this tends to irritate me, the rate if people start talking about the carbon footprint, yada, yada, yada. Well, you can't really talk about carbon footprint if we're not talking about plastic production. So of course, and who knows when this chart was from your book, so I'd have to look back at the date that it references, but essentially, right, a lot of plastic production, so a lot of, fossil fuels goes into our plastic production everywhere. What? I mean, you're right. So but the whole point is we as an entire world use a lot of plastics. So I don't know. And so now let's look at kind of cross-linking reactions. And so cross-linkages, these can connect polymers together. And so sometimes these cross-linkages can also be like the chemistry behind other things like slime and silly putty. So those can be kind of cross-linkages reactions. And so one example, I'll give another example. So this is a demo that I do with people. But if you take sodium silicate, so it's basically this thing, and then you add ethanol to it, it does a similar sort of cross-linkage reaction where it starts replacing some of these oxygens with alcohol groups hanging off of it. And so they're still linked. So that's one example of a cross-linkage. And so this is how I make a bouncy ball. So it has a bouncy ball with sodium silicate and ethanol. And so all those cross-linkages, all they do is kind of give it a different property and kind of makes it almost like a polymer, but it's not a true polymerization reaction.
How are cross-linkages as far as structure-wise compared to you said like slime as an example. Why doesn't it kind of like plastic?
I mean, it just depends on like the properties, I guess. I'm not sure I fully understand the question. Because I guess the difference between like a plastic polymerization is those tend to be like radical mechanisms. So that's what defines kind of like a true polymerization. And then those can be controlled, to a certain chain length. So that's kind of where the manufacturing. The slime and silly putty, that's just more like, I mean, it's hard to know exactly what's going on, but it's just some sort of cross-linkage, right, where it's like chaining up together the silicon and oxygens of the ethanol. So then it starts making like one mass material, right? So they're all kind of linked together. It's just not the same as like a polymerization. I don't know if that makes sense. And so other examples of cross-linking. And so your hair has these disulfide bonds. And so eventually, right, these disulfide bonds can become broken. And so I don't know if anybody has heard of this Olaplex compound for hair. No, oh, some people. But, well, it's relatively new, but a lot of people swear by it, and it's supposed to actually repair these disulfide bonds. So that's why people say that it actually works, and that it works to, reform our disulfide bonds, so then it actually physically, repairs the hair.
So what polymer is that? Would that be like keratin?
Probably, I actually don't know, but I assume, let's look at.
Maybe that's what hair is made out of, right?
Yeah, I'd have to look at the structure. Yeah, I assume this has to be the structure of keratin, but I'd have to look back. All right, let's look at. So then we can look at proteins. And so, of course, proteins are our biological units defined where our monomer is an amino acid. And so this is just our basic structure for an amino acid. So make sure that you grab the 20 amino acid chart if you haven't already. And so I'm just going to pull up my chart over here. And so, right, this is just our basic unit. So they have our carboxylic acid group, of course, and then some sort of R group, which defines them. And then, of course, they all have an amine. And so just kind of go through them and look at them. So there's a couple of things. So first of all, we should point out that glycine, right, has no stereocenter, so that's kind of interesting. Oh, and that's a good reminder. So somebody gave me a homeless titration curve that keeps falling off, so that's yours. Go ahead and claim it.
It does not want to be there.
Yeah.
Paper's topic.
I know.
Just use the marker.
And so let's see, other ones to point out. So what's kind of nice is it does point out the essential amino acids that you would need to consume. And then it has them broken up, right, aliphatic. So this is just like straight chains versus your aromatics. So tryptophan, tyrosine, phenylalanine. And let's just look at, so sulfur-containing ones. So I like to point them out like a methionine and cysteine, since these sulfur groups can be susceptible to attack. So this is how like antibacterial silver coatings work. So that silver will tend to attack the sulfur group, so then that can disrupt your bacteria. And so let's look at some other things. So we mentioned that this carbon has a chiral center. Again, except for, let me pull it up again. All right, our glycine. So glycine is the only one without a chiral center. But let's kind of look at, well, what does this mean, having a chiral center? And so let's just scroll down. So I showed two different forms here. And so when we have something that's chiral, it has what's called handedness. And so we are showing, right, the two different forms of D-alanine and L-alanine. And so kind of there's this whole question of how is life not a racemic mixture. So are you aware that life has chirality? And so let's, sorry, I have too many things.
They're all like right-handed, I think, right-handed chirality.
Yeah.
Is that our-
So it mentions that DNA, RNA are all right-handed, and then the amino acids or proteins are the left-handed chirality. And so scientists are pretty puzzled by this. Now let's see if I can find the article. And I'll probably post this for your reading pleasure, but it's at least interesting to kind of think about and try to understand, oh my gosh, you know, maybe why we have handedness and kind of, well, what would happen potentially, right? And so kind of the mind frame we have to be in is, well, how do we get, you know, chirality from, let's just say, a primordial soup, right? So we're primordial soup potentially, you know, billions of years ago. And then it's kind of asking, well, again, well, how do we end up with this chirality? And so kind of the first problem and why we have chirality, and let's look into how we can make and break, oh my gosh, this chiral center. No, I just want to zoom in. Why does control zoom not work? What is up with this? It was a search, apparently. Okay, I think this works. And so kind of what's interesting, and let's set ourselves up. And so we have our amino acid here. Again, the important part is that we have chirality, and there's a hydrogen hanging off of here that's not shown. And so what this paper is showing and what they do in here is we can say, well, we can convert between the two chiralities between a base. So DBU is just a base, and so all we're saying is we deprotonate it basically, then it has no chirality, so then it becomes achiral, right, once you deprotonate it. And then at that point, it can either convert to the opposite chirality, so in this case going from S to R, or of course, you know, if you reprotonate it, which is what methanol is for, then obviously you can reform the S. So there's our first question, right? Is basically, well, we assume, since again, right, there's energetically about the same, so we would assume that life should be a 50-50 mixture of both chiralities. And so that's not the case, of course. And so this paper is just one of the earlier ones that I'm kind of familiar with. So this, of course, is now from 2007. So a little older. But basically, right, they're trying to see, well, how, again, could we have one chirality in a assumably, right, racemic prebiotic world with, again, probably no or achiral mixtures. So how do you go from achiral to chiral? And so basically, I'll summarize this paper, and what they kind of say is, well, let's say, you know, you stir them forever, and then what if you have selective crystallization? And so that's kind of what they're saying happens. So kind of thinking to Le Chatelet's principle. So let's just say, again, you have 50-50 mixture of both, but it just so happens one of them started crystallizing, and then, you know, obviously this doesn't work well because our hands are chiral, but then what if you had another one of that same, so if I had another right hand and could just keep stacking, right hands, you know, maybe that would lead to chirality. And so that's kind of what people are proposing, that there's some sort of selective crystallization process that happened. And then, of course, once you start crystallizing out, you know, that one enantiomer, then it'll kind of keep pushing to that direction, right? So let's just say the L1 drops out of solution, and then you have this mixture here, so that's going to cause more L to form, and then it's going to keep crystallizing out till it all makes the L. Again, who really knows? And so I think they did some experiments trying to kind of replicate that process. And again, you can kind of read this on your own if you're interested, but basically they just kind of mixed it, and we're trying to replicate those selective recrystallization conditions. And then, let's see. And so this article, this science article, kind of continues this. I think we'd have to check the full version. So this was in 2023 now. And basically, they're still kind of running with that proposal or hypothesis. And this is kind of the relevant part, again, you know, if we think it happens. And so they were kind of looking at this molecule, so this riboamino oxazoline. And so they felt that this crystal could be influenced by the magnetic pole of the Earth's crust. And so they're applying a magnetic field, and we're saying that that kind of led to selective crystallization. You know, again, who knows, but this is just kind of where the field and literature stands. And I did want to make you all aware that there's a lot of literature and time and research devoted to this whole, you know, chirality argument. So interesting to at least think about. All right, so then we'll get into kind of how our amino acids form. And so again, zooming out from our philosophical discussion and back to chemistry. And so we can see that our amino acids chain together by our peptide bond formation. And hopefully this is starting to feel familiar because, oh, look at what we have again. So we have our carboxylic acid group from one of our amino acids can then react with the amine of our second carboxylic acid. So then they will chain by that. And so that's something that, you know, you'll have to do on an exam is like, it'll give you a structure of a peptide, you know, it could be like 3 or 4 amino acids in length, and then you have to draw it appropriately, right? So knowing where your bond forms. But again, it should be feeling good, right? So we have water leaving again, so we have our condensation reaction, and then once again our amide bond. And then just like we've seen before, so this leaves an amine on this side, right, ready to pair up with another carboxylic acid. And then, of course, right, we have our carboxylic acid on this side, ready to pair with another amine group. So just kind of keeping that straight, and then just attaching whatever appropriate R group on there. And so, right, just notice how you can use this as a little template and just change the R group. And so sometimes how they do peptide synthesis in the lab is they have this little, especially if you're trying to make like a peptide and then you're trying to screen it for something. So you might have some sort of bead here, and this might have some sort of indicator to show you if this is good or not. And so then it starts chaining. So, right, we start with alanine, and then in this case, serine. So our dipeptide, and of course, right, this can go on, et cetera, et cetera. And so other things to note are, of course, the abbreviations. I use them here, but I didn't really explain them. So let's go back to our amino acid chart. And so we have both names on there. So we have our single letter code, when biologists use the single letter, and then also the 3-letter code, and then the corresponding DNA codon. So a lot of information there. So I won't make you memorize anything, but you know, they may come up, so use this as your guide. And so again, right, notice how they bond, amide bond formation, water. And then all this is showing too is kind of order matters, right? So we did alanine serine dipeptide, so alanine was first. And then this one is a serine alanine dipeptide, right? So order is different there. And again, our kind of generic chain and being able to identify your amino acids. So right there's 1 amino acid, 2, 3, 4. So just being able to see that, maybe identifying, you know, what these structures were. So that could be something, what amino acids went in. All right, let's take a little break, although I feel like we're going to blow through this chapter today. So yeah, let's take our 10-minute break.
Last night, I realized that my printer reader I was wondering, would it be possible to read between lab and detail? My thing, I have to stabilize my thing, stabilize my most important key to go through.
No, do you want to email it to me now, though?
Who's the?
Yeah, just email it to me and I'll send it over to the printer. Oh my gosh.
Maybe it.
It might be mine. I think if people knew, I thought I swore I put my name on, but
Hopefully it's
I
yours,
do recognize that handwriting.
so you can stop committing suicide.
well, I'm sure I tried my stuff. Yeah, but because I've been trying very hard to put my name on it, because I always forget, because it's not a specific line. Yeah, so I can. Yeah, it's mine. Yeah.
No, ChatGPT doesn't let you look at how to synthesize LSD.
What?
You're gonna go to Arrowhead.
What?
Oh,
What were the words I just heard?
No, Arrowhead knows how to make drugs.
It says it's pretty.
Yeah,
What, to make LSD?
Yeah, it says it used some pretty hazardous.
No.
Oh, they always
Oh, okay, they always say that.
They always like, you probably shouldn't try this at home. Yeah, LSD is just part of me because of the chiral sensors.
Oh, okay.
Not that I have not made LSD.
Wet citric acid.
Yeah. Meth is trivial.
Oh, okay.
Yeah.
But that's not
DMT
something I'm
is
interested
also
in.
easy to
Oh,
follow.
that's what I've heard, yeah.
Yeah,
See,
you
that'd be
just
something
start
I might be
from
more
within.
inclined to try.
Yeah, and then you just methylate it to the
Yeah,
goat.
then, would that form the NN dimethyl, or would that be the 5-MeO? I know there's two different versions, it's 5-MeO, DMT, and NN.
It's actually very good.
I have to double check my
Yeah.
DMT.
Yeah.
Yeah.
This is bad. Well, I started writing it on the board. No. No, after next week. And then I made this table.
Yeah, now that you've paid Aspen, all the register.
Sorry, my neck fell weird.
So I made a scale model of my poster. This did not end up being what this wasn't. I made it to scale, so everyone.
I wish I had, like, a lot of these.
Well, then you got ready. No, it's good, I think the spacing is like, you have to keep you spaced.
Yeah, well, I didn't have enough room to do what, like, I was,
If you remember, it's not a lot.
See, that's where I did my scale model. I did an hour, I spent an hour doing this. The iPad makes it easy, so, I literally could just use it, so, that makes it easy. Well, the title's bad. I think it's yeah, and then I had to, but I think the worst part is figuring out how to like, on my computer, like make a page that would then print out to the side of the hand. Because like, I'm just using like the ruler, like in the whatever, and it's not accurate, the ruler is not accurate. So I like printed it and I was like, it's like 2 inches smaller than it, so then I got it. And then I was like, oh,
Yeah, so like, what I did.
Yeah, I don't know, Sam told me he had quite a bit of issues picking up anything in PowerPoint.
Oh, yes, well, Marissa was the one that printed it out for me, like, it was weird because it had margins and stuff that you couldn't get rid of. Yeah, so it was good, so, I don't know.
at least with Word documents, you can see like change your margin if you like,
Oh,
the minimum, like just on the edge. I think I got it right when it was like I had my picture, I had it on the paper I wanted, and I would add another one, and I'd be moving it around to make it fit better on the page, and change the size.
Did not realize we were supposed to have pictures.
And then it would just like
So,
go off the page, and then
Should
it would
I
go.
print some
And I
pictures and
couldn't
go
find my
on,
finger.
like during your
So I kept
break,
doing
or?
that over and over.
Yeah, probably.
Yeah, okay. Good, thank you.
It's still there, it's just.
So my reference page in the document, at the top, was the last page, and for whatever reason, when I couldn't get it to save the
Did you insert page breaks or did you just hit enter a bunch of times? Because inserting page breaks fixes everything. If you do any,
Oh, because sometimes I just am lazy and just do all letters.
no, if you do that, it messes everything up. It's so bad. I literally had to fix that for free. Literally because why is it doing that? No, so, yeah, no, the page breaks people, it fixed it. And then also like with the figures, where you have to do the text wrapping, you have to change the setting on that.
Oh, I always do that, I always do the type.
Yeah. Yeah. Yeah, so.
Not like,
No, yeah. No, it's, I hate it.
Oh, that's too, that's,
But I guess it worked.
it works. Well, like, I like how you can insert equations in that. Alter the properties and that's for like superscripts and subscripts and all that sort of stuff. So really nice. They have like every single issue.
No, yeah. I met my kids before putting in their figures. So I was like, I'm gonna get six. And then I have to add my figures, so it's like a new exercise. Not putting title.
Once.
I had 8 with mine. So it's like a 6-page exercise. Well, this one, none of my exercises.
That's so stupid.
None
Why
of
would
the
you
articles
do
I
that?
used had a picture of the sprign, but then they talked about it over and over and over. And so my only notes was like, probably included like a picture of this, of the submembrane filter. I was like, find one. I ended up having to store it. Oh, it was one of my original essays. I used it for the information. So then I did it. So I have 2
Exclude.
It's just 5. Like, it was
Mildly stressful because I feel like I'm not taking any line. When there's a line, I can remember it. There's no freaking line.
On your way back to grade school here, we always write our names on it.
I literally have to put so much effort into remembering it, and I thought I did, but apparently I didn't. But there's no line. My brain cannot.
Should put that in the comments. It's just like,
Oh.
this is, yeah.
What line for me?
So I think if it's really
My brain cannot comprehend it, okay? I tried, like, I literally, when I get to class, I try to
I
make sure I
think
write it.
I didn't grade 2 in detail.
But, no, what I'm saying is, hoping to, I'm picking up my years for work during the summer. And I needed Mondays, no, Tuesdays.
Yeah,
Wednesdays,
yeah.
and driver's ed. And then I've been driving there at 9:30 to 11, so it's like right now that I've been working. But, I'm not taking a week off from, from school.
I literally, I think I shared like most of it, because I was like, that's not
And
easy.
usually when I have a week of absolute free time, it tends to make me more crazy, not less
No,
crazy.
literally, I was just writing.
So,
It was like,
After next
then I
week.
changed my title.
I don't know when the final day is.
That's it.
I think, oh yeah.
And it just shows that. Right before they do.
Because like,
But like all the methods are exactly the same. Like you're desalinating water, you're just obviously so. And then I literally, my mom
So
I
wait.
made the same color in the thing, in my thing, so I made this, but this is like lancing the methods that they talk about.
That's the perfect.
Break it up.
Behind the wheel stuff, I don't know.
So his, stuff starts.
Okay.
His classroom portion of his school.
I'm gonna be honest, I
He
don't think
starts at
anyone
6.
is. Okay, who is that?
He starts next week. I may have to have him come to the front drive with yeah, just because it's a big deal also, 9, 11 to 5, Tuesday, Thursday. No, not good time yet. So I think he may have to find his own ride those 3 days.
No, I'm
Because the next week is
I'm
fine.
arguing. Bro, you should have.
There's also,
Dude.
why am I always so busy?
I would not stop laughing. I was dying. Like, I was struggling to not laugh.
And 10 cents.
Those 3 days I have off, when I'm busy that day, It's because I'm like, oh, it's summer, I'm gonna be free.
And then you look at your schedule.
I'm like, no, no, I did it to myself. But it's, I usually do actually prefer to be kind of busy, because when I'm not busy, I get very frustrated right away. Because I'm like, what am I doing? Why am I here?
I was upset about taking classes this summer. So I remember last year, I took the whole summer off. But that just left me like marinating in all the drama of my family and not anything else.
I just always felt like I'm just,
And not doing anything on top
I have
of
a
it.
very hard time with not having things to do and not being productive.
Yeah, I like it.
Sometimes it doesn't work.
Doesn't mean I'm the most productive.
Well, like what I need is that one day of absolutely nothing and then 2 weeks straight of something. And then 1 week of nothing. Is there any more than that? And I'll start to like,
So you just put it on the back of the box and then
I went everywhere last summer because I didn't have anything to do
yeah.
on top of it. So it was like, not really doing the only thing I could focus on was everything that I couldn't control.
Yeah.
yes, you can.
I don't know. That is awesome.
I think you do.
Yeah, I was about to say, maybe you could just write an
Well,
L
not for me.
somewhere, but that's like
Well, again.
not really to this two X's.
No. You're not gonna go, that's not how that works. How many people have met you on the app?
Sorry, that's hilarious.
Dieting makes me
Well, I mean, it's spelled
Or
wrong.
not this weekend,
It's
next year.
just the wrong word
And I
entirely.
couldn't go.
When
She's
you're
leaving
so wrong.
Thursday morning. I work on Thursdays and there's no.
What is your schedule like in during the summer? Thanks. Such description.
I can't
Well, I
spell
have,
this out loud. I'll say it.
you stay on that.
That's in July.
Oh, yeah, I think it starts with that.
Well, because we were. Well, we're taking our break.
I wrote the entire thing.
I have a.
Well, I work for it.
Really what I heard was I woke up. My alarm went off and then I didn't get out of bed. Now I don't want to die. Like I can make that's it. Get ready. I was still.
Not allowed to fish on it or anything like stuff like that, so it's like.
I look over and I see.
Yeah, there's a lot of little clips and stuff. Is there, I remember as a kid, the first spot to go to on the art, it was like still working, there was a higher cliff back jump forward. Oh, yeah.
I don't know, like, well, we got one more again. I texted her, I can't get to the colors. Well, I think that was like around that time.
I emailed you a couple documents, please, thank you.
Well, no, it's because I have like. Well, no, I'm not talking about that.
All of them.
That's why,
All the ones that were on my desk.
Sounds like pretty much the only option he's got.
Sorry.
It wasn't, but it
This,
Too big?
yeah, about twice as big. I might have gotten too small.
Okay, I'll print it again.
Okay, and it should be, each of those quarters should be 1 page. So I measured it so that 1 page is 1/4 of this.
Oh, okay. I just did a 4x4, but I can reprint it.
Cool, thank you.
I can't get to the color. I have to ask Nicole how to use the color printer. Because I have to send it to the other color printer. All right, let's continue. Can't keep what?
Can't keep mingling.
We're almost done with this chapter. And then you can mingle. And so now we start getting into kind of the primary, secondary, tertiary structures. And so this is probably from biology. And so our primary structure is just our sequence of amino acids. And then this is just to give you context. So of course, right, all these amino acids start to get pretty lengthy. So insulin is 51 amino acids. Yeah, myoglobin is 153. Keratin, which we were just talking about, so 204. Hemoglobin, that transports our oxygen. That's 574, so these get pretty big. And
How do you categorize that there's 51? Is that like it equals to 1 mole of it, or?
so I'm not sure if they use like moles still with insulin, but yeah, that's kind of the thought that, right, you have 51 amino acids make up your structure of insulin. But then, of course, right, it gets like more complicated than that, right? Because first we just have the sequence of amino acids, but then they start to fold, right? So then you have your secondary structure, and let's just pull up like Wikipedia.
I know, I was thinking that.
I'm like, oh yeah,
I understand
alpha.
this
I'm like, oh yeah, natively she alpha.
Yeah, so then, right, you have, of course, just and this is a nice breakdown as well, right? So we have our primary structure, like we said, so just our chain. But then the secondary structure is, well, how, does that chain start to fold and twist? And so that's just called our secondary structure. And so kind of the common ones are these alpha helixes, or kind of spirals, or these beta sheets that tend to turn like that. And so those are like the main ones, but then there's, other ones, so these omega loops. I don't know if they have a picture. Yeah.
You gotta use your imagination.
Apparently it resembles an omega, but it'd be nice if they had a picture. And of course, right then, that secondary structure goes into the overall tertiary structure, so our overall shape of the protein, of course. So tertiary, so now just our whole 3D shape. And then, of course, right, quaternary is just where is our protein in relation to other proteins. And so, again, for our purposes, we just kind of want to appreciate this, but then also understand that probably, so this, gives rise to our, we'll say, actions or catalytic properties. And so what I mean by that is we can think back to any of those different catalysts that we had looked at. So let's just look at the nitrogenase enzyme. And so, right, we have nitrogenase, and as a chemist, at least, from my perspective, we usually just tend to look at kind of the active site, which does kind of ignore, right, all those secondary, tertiary structures that really give it kind of its properties. And so that's the other thing too, right? We don't fully understand, why the tertiary structure gives it that overall function. Again, the chemists focus on the active site, but obviously, that's not the full picture. And here we go. So right here's, again, kind of our active site of nitrogenase. We kind of mentioned this before with these iron-sulfur clusters, but again, right, we're ignoring the whole secondary, tertiary structure of nitrogenase. And let's add, so I forgot quaternary, well, let's at least add that, so quaternary structure. So then that is, right, the 3D form and the interactions of multiple polypeptide chains. And so kind of, bioinorganic chemistry would get more into this as well, like looking at the active site and maybe somewhat trying to get into our other higher level structures. And so, of course, other important polymers are RNA and DNA. So these would be our polynucleotides, right? So made up of different nucleotides. And so our nucleotide is just our sugar, and then a phosphate group, and our base. So there's our DNA and RNA. And then, of course, we have are 5 bases that occur in DNA and RNA. And it mentions, so thymine is only found in DNA, and then uracil is in RNA. So DNA, RNA, I think you probably know that from biology. And so then we want to kind of identify the pieces of our DNA. And so of course right here is our phosphate group, so just identify that. And then right, our little ring here is our sugar. And then attached to our sugar is our base here. So there's our unit, right, phosphate sugar base, and then that just repeats, and now our sugar is coupled to another phosphate group, and then that's connected to our sugar base again, and then that just repeats. And then so forming our double helix then. So what's responsible for our double helix? Yeah, hydrogen bonding. So hydrogen bonding, super essential. And here it is. So just kind of, right, reorient yourselves. So we have our amine from 1, and then that's causing, right, our hydrogen bonding interaction from our oxygen over here. And then our another NH over here. So again, hydrogen bonding between that nitrogen and hydrogen. So our hydrogen bonds, and they're in the dotted line, so just to emphasize that. And so now we're also answering why these certain base pairs pair together. And so you may know this from, again, biology. So right, A and T are paired together, kind of both with the straight looking letters, right? So all straight lines, so we know they pair together. But why these two pair is going back to the hydrogen bonds. So these ones have 2 hydrogen bonds across, so right, they match really nicely. And then scrolling down, so C and G, which of course, right, is our abbreviation for cytosine and guanine. So those pair. And so notice these have 3 hydrogen bonds now. So that's why those are paired up. And usually the trick is, right, they're both kind of curvy looking letters, so you know they pair together. But again, the chemistry is our hydrogen bonding. So there's our amine again, hydrogen bonding. So hydrogen bonds. So those have 3 hydrogen bonds between them. So that's, yeah, that basically forms, right, your ladder then. So that's what's able to kind of form the ladder so that they can pair up and make our ladder structure there. No, it's just whatever sugar, but yeah. And so now here's just our larger picture of what's going on. So again, seeing, right, our double helix forming. So A and T paired, again with our 2 hydrogen bonds. G and C, 3 hydrogen bonds. A and T with 2 again. And C and G with 3. And I guess we'll add to this, so right, A and T pair, but then sometimes it's U. So depending if it's RNA. So that's the only oddity you have to remember. And so then, let's see, what do we have left? So now, how do we code for different amino acids? And I'm sure some of you can help me make sure I don't mess this up. So how do we code 4 different amino acids. And so we have, right, DNA, and then it pairs with its complementary pair. So if we have 5' for our DNA, so A, C, C, T, C, G, A, G, and then 3', then our corresponding would be T, and then you just pair them, so right, A goes with T, then C with G, T with A, C, T, C. Sorry, scrolling around. So then let's go over, where is it? Oh, so page 9 then has all the corresponding kind of start-stop cheat sheets here. So let's try one. So let's see. Let's see what this one corresponds to. So we have 5', CTC, GAG, and then what's our stop codon? And then GT, right? I think that's a stop codon. And so then we can just figure out what amino acid it pairs with. So we start with the first 3 code. So CTC. So going down, so I'll look at this one because it has C, so C, T, C. So then that corresponds with leucine here. So then we can identify that. So then this is leucine. And then we find GAG. So I'll use this one. So GAG, so glutamic acid. I think it's just supposed to be a stop codon, but I don't know if I wrote it wrong.
Well, that one, the last one, that one only has 2.
Yeah, all the stop codons are 3.
Well, all the codons are 3. You just gave it a mutation.
Yeah, I think I must have a mistake in my notes because I just have like, so I'm not sure what this is supposed to be, but I think it's supposed to be a stop codon. And I was trying to figure out what my note was going for. Yeah, now I'm confused. Let's just make this TGA. I think that's a stop codon. So hopefully that was stop, so TGA, so corresponding to our stop codon.
Why don't we even start then?
I'm not a biologist. That's the answer to that.
Does it mean that we do something better than you guys do? Is that what you're saying?
You know, I'll concede in this one.
It's on the second one.
All right, let's see. Let's go back to starches for a second. So now we want to look at starches. We might go back to DNA later. So we'll look at starches. So starches are just defined as polymers of sugar. So similar. So now we're going to have another condensation reaction. Surprise, surprise. And so then we have our two different forms here of our sugar. And so it's just looking at how they add. So our alpha form, let's put this side by side. So now we're looking at alpha versus beta. And so we're looking at these OHs. And so in the alpha form, they're on the same side of the ring. And so this one, so kind of both pointed down. And then in the beta form, they're on opposite sides. So this one's pointing down. This one is pointing up. And so then when it adds, it can be in different kind of geometrical relationships. And so now we're just showing kind of our alpha connection here, right? So this is our alpha glucose. So both on the same side, and then it can add. So now we have our alpha linkage there. And so still pointing down. So that's just showing us how it can add. And then again, condensation reaction, right? That OH on our carbon leaves. And then hydrogen leaves. This is doing the action, right? So notice electrons come from this alcohol group and attack our carbon there. Sorry, I'm trying to think what else we should cover. Let's do a little practice, actually. Oops. Oh, the other thing, so I think I was trying to point out, so glucose and galactose, so these are pretty similar in structure, but not. And so this was just going to point out that these two are isomers. So looking at this OH group, so notice that it has different positions. So this one is equatorial versus this one is axial. So these are just isomers or diastereomers would be another term. So just basically different stereocenters. Let's see if we can practice. So one thing you will have to do is draw an amino acid chain. So see if you can do that with these four. Oh, no, I haven't, sorry. How are drawings going? So let's check them. So we're going to pull up our amino acids again. And so it's really just piecing them together, right? So we have our glycine to start off with, and we can always use, right, just our little mini structure. So I'm just going to start off with carbon. So our amine group. So right, just drawing the amine, and then I'm just drawing our 2 carbons kind of in our chain here. And then glycine doesn't have an R group, right? So normally you would put your other R group there. It's just a hydrogen. And then I'll switch colors, so then let's look at, should be methionine, I believe. I don't know my amino acids. So met, so then we can do this one. So right, this is our R group. So that kind of maybe helps. And so now we need to couple it to the nitrogen. So let me pull our structure back up. So right, we're coupling it to this nitrogen, then carbon, carbon. So carbon, carbon, H. This is our R group, so now I can draw this CH2, CH2, S, CH3, and carbon, carboxylic acid. Then we're ready for the next one. So phenylalanine, so now right, connect it to the nitrogen again, 2 carbons, nitrogen, carbon, carbon, so now R group on there. So this methyl group, benzene ring. So something like that. H, oh, I forgot I left off my hydrogens here. C, double bond O, and then finally we're ready for the last one. So glycine. So then we end with this one again. So same thing, nitrogen, carbon, carbon. Now we leave our carboxylic acid group on there, and then just fill in our R. Any questions about this one here, how to draw it, how to connect it?
Yeah, kind of. So with methane, how do you know to draw the carbons just going down with the sulfur like that, just going down just like that?
Just because, right, this is our R1, and then I guess, are you confused about like why it's straight, or just, because I just drew it straight, so it still has like the 3D.
So with our R in this scenario, is that kind of the NH, just kind of where that's located? I'm a little confused trying to figure out how to get that from the amino acids.
Yeah, so your R group is always whatever is hanging off of it. So like this thing for alanine, right, this whole thing would be your R group for tryptophan. Right, this is your R group for serine. So it's whatever hanging off of your carbon that's attached to your amine, right? So you have this, it's basically attached to your chiral center carbon. And so it's kind of easier if you just take this like template roughly, right? So then you can break it down where you just have amine and then carbon. This is R1, then so carbon, carbon, and then you start your new one, right? Amine, carbon with your R, whatever R group, next carbon, and then you just kind of keep repeating. I don't know if that helped. Yeah, because you got to kind of see, well, it's making that connection there, but then you have carbon, carbon, right? And so this is a carbon, right? That's that carbon. So I just like to draw the R group hanging off of it for simplicity. So it kind of, makes it look like a simple straight chain. Obviously, I'm not showing the 3D structure here, but.
I'm just also just gonna like to look at especially the methane, because it's rotated differently.
Yeah.
So you have that rotated differently, but you can see on the table, so I'm just kind of trying to figure out.
Yeah, so these are all right, carbon, carbon. So this is your first carbon, carboxylic acid, carbon, and then, right, so remember glycine has no R group, so the R group is H. Isoleucine, right, this is your R group. R group. Yeah, so I would just, let's kind of do that. We'll sketch our, right, generic models. So we always have kind of the same format, right? Our amine, then carbon, carbon with our R group, H, double bond, right? And then it starts again. Amine, carbon, carbon, right? Now your R, whatever, R group again, carboxylic acid, then repeat.
Is it still an amine or is it an amide now that it's bonded?
Yeah, so then it'd be an amide, correct, now that it's in the bond. And then whatever's at the end is either carboxylic acid or just continues. And R group. Yeah, so then you're always losing, plus water on all of these, every single condensation reaction. Yeah, and taking a peek at your exam. Yeah, you'll have something of kind of examining a structure and identifying errors. Yeah, so it shouldn't be too bad. And then other things you'll have to do is like figure out like what substances form your copolymer. Yeah, there'll be a little bit of IUPAC naming. All right, we will end here, and then I will see everybody at 1:30 in our lab. So.